In the first section of the course, you heard about the impact of nanoscience and nanotechnology in areas like computing, communications, and imaging. We're now ready to leave the physical world and enter the biological world to discuss nanoscience in the world of biology, and as well the applications of nanotechnology to medicine. Biology fundamentally operates at the nanoscale. The basic building blocks of life are all around 1 or 2 nanometers in size. So things like amino acids, sugars, lipids, even DNA molecules are all nanoscale objects. Only slightly larger than these molecular building blocks are the vast world of molecular machines and the messengers that drive cellular function. Now, historically, the cell was thought of as having a few major organelles that did most of the work. For example, the cellular mitochondria, which are the power producers in the cell, they're about 1,000 nanometers, so a little too big to be nanoscale. But we now know that there are thousands of smaller different molecules that run around inside cells, and these molecules allow cells to live and replicate. So, for example, proteins are synthesized by molecular machines called ribosomes. And these ribosomes, which perform a very complicated function, are only 25 nanometers in size. As well, energy for the cell is produced by an enzyme called ATP synthase. And this enzyme, again, a very complicated machine that has a very important function, is about 10 nanometers in size. And the cell membrane, which holds everything in the cell in place, is typically about 10 nanometers too. So you can see that this idea of biology really being nanoscale spans many, many different aspects of biology. As well, certain self-replicating organisms can also be nanoscale, although just barely. So certain viruses can be around 20 nanometers, others are about 400 nanometers, but then the very smallest bacteria are about 1,000 nanometers, so not really nanoscale. Now, as you might imagine, nanoscientists derive lots of inspiration from biology. We look at the complex world of biology and we marvel at its ability to self-organize, and we try to recreate what we see with the tools of nanotechnology. And it will be important for us to understand some of these nanoscale activities of the cell so that we can understand disease mechanisms and the action of nanodrugs, and as well the logic behind nanodiagnostic approaches. So in order to do this, we need to take a look at the biomolecules that are responsible for all of the action within a cell, and we need to understand how they're structured and how they function. And then we can explore how these biomolecules can be put together with nanomaterials to do something, let's say, like advanced drug development or help with the invention of new diagnostic tools. Our next 2 lectures will combine discussions of basic biological processes with nanotechnological applications. And then we'll move on to some discussions that are more focused, and they'll be focused on applications of nanomedicine. So we'll start in this lecture by looking at a new type of diagnostic testing based on gold nanoparticles that has very exciting potential. What is so exciting about this is that this is a completely new type of testing that can read out a variety of DNA mutations that give rise to genetic disease or drug reactions. And this type of testing relies on a very simple measurement, the change of color of a nanomaterial. And this was one of the first nano-enabled diagnostic systems to come onto the market. The discovery that this diagnostic approach is based on was made in the late 1990s at Northwestern University in Illinois. A group there in the chemistry department discovered how to attach small DNA molecules to gold nanoparticles. Now, how did they do this? Well, they identified a type of atom, a sulfur atom, that was very good at sticking to gold. So they then attached a sulfur atom to the end of a sequence of DNA that they synthesized. And with these DNA nanoparticle complexes, they showed that they could read out small changes, or what we call mutations, in very long DNA sequences from patients. And the detection of DNA mutations, as I'm sure you know, has tremendous relevance to the diagnosis of genetic diseases like cystic fibrosis. Genetic diseases are caused by very small changes in an individual's DNA, and these changes then get passed down from generation to generation. The gold nanoparticles, again developed at Northwestern, were able to scan DNA molecules for these changes. And if a problem with the DNA was identified, the particles changed color and they turned from red to blue. Now, how can something as complicated as genetic disease be detected with a simple color change? Almost seems way too good to be true. So let's take a close look at this so we really understand how it works. So in order to really have a complete picture of how this type of diagnostic approach works, we need to understand three different components that are coming together. A gold nanoparticle, a small DNA molecule attached to the particle, and the DNA genome that's being scanned. So let's start with the DNA genome so that we can understand the challenges involved in genetic disease diagnosis. As I'm sure you're aware, DNA is the molecule that stores all of the information our cells need to function. It does so by programming information in a biological code. And so if we zoom in at the molecular level, and keep in mind that this is also the nano level, we find that DNA is a double-stranded molecule. If you look closely at the structure in the figure, you'll see that there are two strands that are wound around each other, and they form this very famous double helical structure that I'm sure you've seen before. Now, why does this helix form? If all structure is to be present in DNA, why do we get this helical structure? And how does it carry a code? Well, I'm sure the answers are familiar to you from introductory biology. The key feature of DNA that is relevant to both of these questions is that it contains 4 different building blocks that we call the DNA bases. They each have 1-letter abbreviations, either A or G or C or T. And don't worry too much about these names, but the 1-letter abbreviation either stands for adenine, guanine, cytosine, or thymine. All 4 of the DNA bases have different chemical structures. So you'll see that A is colored red, T is yellow, G is blue, and C is green. And if you look closely at these structures, you'll see that they have not necessarily different kinds of atoms, but atoms that are arranged differently. And so each base has a unique structure, and we'll talk about why that's important in a few minutes. Now, the helical structure of DNA results from specific attractive forces between bases that dictate what partners they pair with. Now, why is there this pairing? Why are these strands attracted to one another? It turns out that hydrogen bonding is what's key in holding the structure of DNA together. Hydrogen bonds are bonds that actually hold molecules together kind of loosely, but with a high degree of specificity. Now, these types of bonds, not surprisingly, they're called hydrogen bonds, and they involve hydrogen. So you have to have as one part of the partner a hydrogen atom, and then as the other partner, we usually find things like oxygen or nitrogen. And it turns out that all of life, all of the molecules that are wrapped up in our cells are held together by the attractive forces of hydrogen bonds. So, as I said, this is a loose type of bonding, but it's very specific, and it's enough to have us live and breathe and do all of the things that we do. And as you look more closely at the structure, if you now can pick out a hydrogen bond, you'll see that the A moieties bind to the T's, and they have 2 hydrogen bonds. The G's bind to the C's, and they have 3 hydrogen bonds. So there are these different levels of bonding in the different base pairs. So these attractive forces between different bases cause the strands to pair up. This is why DNA helices form. And the specificity here is amazing. So you can take a piece of DNA that has 100 units within it, and if you introduce the complementary sequence where for every A you have a T, every G you have a C, and vice versa, those two DNA molecules will line up perfectly, and there won't be any mistakes. It works every time. And even more amazing is the way these bases are arrayed is what codes the genetic information required for life. So the pattern of bases that are found in a DNA molecule gives a cell a full set of instructions, so it knows exactly what to do to keep itself alive and healthy. Now, I've shown you the structure of DNA and pointed out its key nanoscale features. But how do we know that this is exactly how it looks, and how was the structure of DNA originally characterized? Now, to answer this question, we have to go back to the 1940s. At that time, members of the scientific community were aware that DNA was most likely the molecule of life that carried genetic information. They also knew that DNA included different amounts of the 4 units, A, T, G, and C. But nobody had the slightest idea of what the molecule might look like. There were proposals, there were models drawn on a piece of paper, but it was all really guesswork. And you have to keep in mind that when this question was being answered, this was long before we had the ability to image molecules at the nanoscale. So 2 men, James Watson and Francis Crick, ended up being the ones that eventually did solve the structure. And they got a start on getting that structure using stick and ball models. And they were working on this problem in the 1940s at the University of Cambridge. And you'll see in the picture that you're viewing a depiction of their model, then 2 of them working on it. And it may seem funny to try to solve something as complicated as DNA structure by building something out of wire and trying to get it to work and come together. But if you're thinking about something chemical, it's actually quite a valuable way to build up a structure because you know what the bond angles and bond lengths can be, and you can eventually put together something that makes sense. Now, at the same time, other scientists were using experimental methods where they were trying to directly characterize the structure of DNA. Rosalind Franklin and Maurice Wilkins, who were also in England, but they were at King's College in London, used X-ray diffraction to understand the physical structure of the DNA molecule. Now, when you shine X-rays on a crystal of DNA, the invisible rays bounce off the sample. The rays then create complex patterns on photographic film, but it would take hours and hours to capture a single image. But then by looking at the patterns, it was possible to get important clues about the structures that made up the crystals. Now, Watson and Crick were building their models, but they were having difficulty getting the strands to match up properly. Every time they would stand in front of their model and they would try to bring the 2 strands together, they just couldn't make it such that it was stable. They knew it would just fall right apart. And it was as though they were trying to solve a puzzle, but they weren't using the right pieces, and they just couldn't get the whole thing to work. Now, a major breakthrough came when they saw Rosalind Franklin's famous photograph 51. This was an X-ray diffraction pattern of a DNA sample. And you'll notice that it has a fuzzy X in the middle of the molecule. And this is not actually a direct image of DNA, but the diffraction of X-rays bounced off of the DNA show a pattern. And this pattern indicates that there's a helical structure there. And so that told them that the structure of DNA was a helix, and that it had a certain periodicity that told them about the spacing between the bases. And it was this information that allowed Watson and Crick to finally get the structure right. They published their structure of DNA in the journal Nature in 1953, and this paper was published alongside of a paper from Rosalind Franklin, another paper from Maurice Wilkins, describing the experimental data that they had collected. Now, Watson, Crick, and Wilkins received the Nobel Prize in 1962 for their discovery. Unfortunately, Rosalind Franklin died in 1958 of ovarian cancer and thus did not live long enough to be recognized for her very important contribution to solving the structure of DNA. So now that we know something about the nanoscale structure of DNA, let's think about the much longer pieces of DNA that are present in a cell. So there are 6 billion bases in the DNA found in every cell. Each base takes up about 0.3 nanometers in a linear sequence. So that means for every 10 base pairs, you have about a nanometer of DNA. 100 base pairs give you about 10 nanometers and so on. So if you think about it and you add up the numbers, that means that there are 2 meters or over 6 feet of DNA in every cell if you were going to stretch it end to end. So the DNA in just one of my cells, and there are trillions of cells in my body, if it was stretched out linearly and vertically, it would be taller than I am. That's just one cell's worth of DNA. So if you took all of the DNA in all of your cells and laid it out end to end, it would actually stretch to the moon and back about 130,000 times. It's amazing to think about, and I really don't suggest trying to see if I'm right. You'll have to just take my word for it. As for one thing, it would be very hard to get the DNA back in your cells after you took it out, and it would also be very expensive to fly back and forth to the moon every time to check that. So hopefully, again, you take my word for it. But it's really amazing to consider how much DNA it takes for us to function. And the reason that we can fit all of this DNA in our cells is that it is condensed into tiny particles, and it's not present in a linear form. In the figure that you're looking at, you can see how the DNA of an X chromosome, which has 155 million base pairs, but is only about 100 nanometers in size, it really needs to come unraveled for the linear form to be the least bit visible. So now that we have an understanding of the structure of DNA, let's talk about the role of DNA in genetic disease. And then we'll be able to look at how nanomaterials can be used for DNA-based diagnosis of genetic disease. Now, patients affected with a disease called cystic fibrosis typically have mutations in a gene that dictates how well their cells can deal with salts. For example, the most common mutation causes 3 bases to be missing from a sequence that codes for a protein that is essential for cellular function. This causes mucus to accumulate in the lungs, and serious bacterial infections result that then interfere with breathing. This is a very serious disease. Now, let's come back to the gold nanoparticles that can sense these mutations. How do they do it? As you now know, DNA consists of 2 complementary strands in the cell. If we want to target a particular sequence of DNA to diagnose a disease that would result from mutations in that sequence, we can take a man-made strand of DNA and design it to stick to the region of the mutation. And how well it sticks will tell us whether the mutation is there or not. Now, in order to detect the cystic fibrosis mutations, 2 different types of gold nanoparticles can be made that will line up specifically with the region of mutated DNA. And this is depicted schematically in the graphic that you're looking at, so hopefully you can see what's going where. If the altered DNA sequence is present, the gold nanoparticles will bind to it and they'll come together. And when this happens, the nanoparticles will change from red to blue. And it's that color change that tells us that the mutant sequence is present. Now, that may sound a little strange. One material, a gold nanoparticle, changing color because it's binding to DNA. So I want you to really believe me that this is what happens. And so I'm going to do a demonstration of this so that you can see for yourself how it works. Okay, so what I have here is a solution of gold nanoparticles. And as you see, they're just suspended in solution, nothing really hanging around in the solution. These are all very soluble and stable. And I've put some of that gold nanoparticle solution into 2 different test tubes. And what we'll do is we'll add DNA to one, but not the other. And we'll see what happens in terms of the color change in the tube where DNA was added, and then we'll compare it with the one that was just left alone. So here's my solution of DNA. So just a clear solution. Again, DNA is completely soluble, doesn't really have any color of its own, so this just looks like water. And so I'm going to take a few drops, add it to one of our gold nanoparticle solutions, and then you see what happens. So you can see that right away, the solution is changing color. And again, we started out with red, I added something that's clear, and now you can see that this guy is turning kind of bluish purple. So that is the color change that's affected by gold nanoparticles brought together by a complementary sequence of DNA. So that's exactly the type of effect that's used to pick up on the presence of genetic mutations. Now, if we step back for a minute, this is kind of curious, right? We are talking about gold nanoparticles. We all know what color gold is in its solid form. It's gold, right? So now, why did I start with one solution that was red, and then I was able to change it into a blue solution, and this all of a sudden starts to seem kind of confusing. So if you happen to have two rings that are made of gold, and you bring them together, absolutely nothing happens, right? Gold in its metallic state really doesn't ever change color. But here we're not talking about macroscale gold, we're talking about nanoscale gold. And when nanoscale gold is dispersed in solution, it has a very different set of properties. And so that red color is related to the nanoscale size of these particles. And this is a great example of how a nanoparticle or a nanoscale object can have a completely different set of properties than the macroscale version that maybe we're used to looking at. So this is a key point that illustrates why nanomaterials can have unique utility. This color change that we're talking about, and the fact that the gold nanoparticles in solution start off red to begin with, arises because of the interactions of the particles with light. And if you think about light and the waves that make up light, those waves are on a length scale of nanometers. So we talk about the spectrum of light, and the unit that we describe that in is always nanometers. So the fact that you have light with these oscillations that are nanometers, you have a particle that has a set of dimensions that are about on the same size scale, there is an interaction there that is unique. Again, macroscale gold that doesn't have anything nanoscale going on doesn't really interact with light. It just reflects light. But here, with these tiny gold nanoparticles, we have electromagnetic waves that are dispersed on the surface of the particle. And there are highly delocalized electrons there. And so the wavelengths of light and these waves on the surfaces of the particles interact, and they dictate what color the particles appear to be to the human eye. So gold nanoparticles that are surrounded by water and not close to other particles appear to be red. When the nanoparticles come close to one another, they appear blue. So again, it's the same material, but it just has a different distance separating it. And the fact that the absorption of this material becomes blue reflects the fact that they become electronically coupled. What does that mean? Well, it means that their electrons actually start talking to one another. They know that they're there. And then that perturbs the surface waves on the nanoparticles. And it's this perturbation of those surface waves that gives us the color change that we see when nanoparticles line up on a strand of DNA in the presence of a disease-related mutation. So now you understand how gold nanoparticles can be used to detect the DNA mutations that cause disease. And you're probably wondering, is this something that can be done now, or are we talking about something that is years away from being available? Well, it turns out that nanoparticles are actually being used today for exactly this application that we've just gone over in hospitals all over North America. A company called Nanosphere that was launched out of Northwestern University, where all of this research began, has taken the gold nanoparticle technology and developed an instrument that can automate its use. So me squirting my solutions here on a bench, we wouldn't necessarily want somebody to be using gold nanoparticles that way to do something as important as diagnose a disease, but the same concept has been packaged up into an instrument. So in a testing laboratory, a technician can take a sample of blood. They extract the DNA from the cells, so they break the cells up and they take the DNA out and they purify it. And then they inject it into a cartridge, just a small testing unit that can mix it up with the nanoparticles. And then this mixture is put onto an array. And what the array is doing is picking out a pattern of blue spots or pink spots that allow us to know what mutations are present in the sample. And this is important because there are many different DNA mutations that cause cystic fibrosis. And so it's important to be able to look at many sequences at once, hence the need for lots of different spots for the same test. We wouldn't want to miss, let's say, a very rare mutation and miss the diagnosis for that reason. So being able to look at lots of things at once is very helpful in that regard. Now, this type of testing instrument, in addition to being used for cystic fibrosis testing, can also be used to see if patients have mutations that make them sensitive to certain drugs. So genetic factors have much to do with why individuals react differently to the same drug. And there's a nanosphere test now available that allows physicians to assess this genetic predisposition before writing a prescription, so before they give a patient a drug that they're going to have a bad reaction to. A very powerful capability. Things like viruses and as well bacteria can also be detected with the Nanosphere instrument. Different viruses and bacteria have different genetics, and so the same principle can be applied to diagnosis of viral infections. So it's very clear that this technology has tremendous utility. And as I mentioned at the beginning, this is one of the first nano-enabled diagnostic systems to come onto the market. This advance is the first of many, and in a few lectures we'll discuss other types of diagnostic approaches that are a little bit earlier in their development, but have just as much promise. And so we'll come back to this idea of looking at nucleic acid molecules, using nanomaterial to do very sensitive and specific detection, and we'll look at a variety of different approaches that can achieve this, again, all using nanomaterials. So in this lecture, we've covered some key concepts that will help you to understand nanomedical advances and how they work. You've now seen an example of a nanomaterial, a gold nanoparticle. This has come up in other lectures, but now you've seen how these materials can scan DNA for mutations. We also discussed a special property of nanoscale objects, and that's the interaction with nanometer light waves. And this gives nanomaterials like gold very different colors than their macroscale counterparts. And you now know that this special light-changing capacity can be used to diagnose genetic disease resulting from DNA mutations. Now, in the next lecture, we'll talk about nanomaterials that are given special functions by the attachment of a different type of molecule that's also nanoscale, a protein. So we'll look at the structures of proteins, go over their functions, relate how the functions of proteins can be used to manipulate nanomaterials or to make them more powerful. See you then. Everyone knows that protein is an important part of our diet, but do you know why? Well, our bodies are actually 20% protein, and it's this material that allows us to think and run and do all of the other things that we do. Protein is a very important part of our diet so that we can make the molecules that we need internally and keep our cells running. Just like DNA, which we covered the last time, proteins are a major class of biomolecules that can connect biology very directly with the world of nanotechnology. What we'll do in this lecture is to look at some of the basic features of proteins and see how these features have been used to manipulate nanomaterials or make them useful for applications in nanoscience and nanomedicine. Proteins are a type of cellular molecule that, unlike DNA, have a wide range of functional roles and structural motifs. So when we talked about DNA, we talked about its very regular structure and the fact that it really only does one thing. It stores genetic information. Proteins, on the other hand, have many, many different functions. So they drive all of the chemical reactions that occur within cells. They provide structural support to cells and tissues. They're also the gatekeepers of cells, controlling what molecules are allowed in and out. And proteins are also the key players in our immune system. They're able to recognize foreign cells like bacteria and viruses and target them for destruction. So before we talk about how proteins and nanomaterials can interact, let's consider the nanoscale structure of proteins themselves. These molecules are composed of small building blocks that we refer to as amino acids. And when we call them small, what do we mean? Well, they're no more than 1 nanometer in size. So the picture that you're looking at shows the structure of tryptophan. This is an example of an amino acid you're probably familiar with because of its reputation for making us sleepy after we have Thanksgiving turkey. That's not its major function, but that's one of the things that it can do when it's floating around in cells. It turns out that there are 22 versions of tryptophan that serve as building blocks in proteins. And they're all slightly different. Some are more greasy or water-friendly, or they may have a molecular charge. The part of the amino acid that you're looking at that's gray is what varies from one amino acid to another. The rest of the molecule stays constant. And the ordering of the amino acids within a protein can have a strong effect on structure. Now, how is the sequence or the ordering of the amino acids within a protein determined? Well, this is what the information within DNA is used for. It's turned into a protein code that gives cells the molecules they can use for all of their various functions. The first glimpse of protein structures at the nanoscale came around the time that DNA structure was first characterized. It was much harder to interpret the protein data, though, because unlike the simple double helical structure of DNA that is very uniform, it turns out that proteins have structures that are highly variable. So let's take a look at a protein named hemoglobin. This protein carries the oxygen from our lungs to other sites in the body, or if you happen to be a fish and you're tuning in, it carries the oxygen from the gills to the rest of the fish. And this protein is roughly 6 nanometers across. You'll notice that there are curly helical sections of the protein structure, but it's not a straight continuous helix like DNA. There are sections that are not helical that are interspersed, and these can be quite disordered. And this is one of the things that makes structural characterization of proteins extremely difficult. Another challenge was that there were 22 different amino acids that could be present within the protein, and having so many building blocks made it much more challenging to fit everything together. But a breakthrough was finally made in this area in the 1950s, which, as you'll remember, was a decade after DNA structure was characterized. 2 researchers working at Cambridge University, Max Perutz and John Kendrew, spent an entire decade trying to deconvolute protein structure, and they were finally successful in 1957. And they received the Nobel Prize in Chemistry in 1962, and you'll remember probably this is the same year that the Nobel Prize in Physiology and Medicine was awarded for the structure of DNA. Now, why are the structures of proteins so variable? And we think this is connected to the fact that they perform so many different functions within the cell. Some proteins have evolved to very specifically recognize other proteins, or even DNA or other molecules, and so their structures are designed to find and bind partners. The helical sections that you find in proteins are very rigid, and so they're good at grabbing chemical functionalities on other molecules. And this is where different amino acids come in. The ones that are good at making hydrogen bonds can reach out and make contact with other molecules. The linker regions that you saw in hemoglobin are quite flexible, and they're good at allowing a protein to change its shape to give off a signal. Proteins are able to change shape, and they can tell a cell whether the cell is cold or it's hot or it's hungry, and it can give off these signals so that the cell can do something about it. Other structural motifs that are found in proteins can provide binding pockets where chemical reactions can occur. So now that you know what a protein looks like, and you have a feeling for the highly variable structures of proteins, let's discuss what proteins do and connect their functions with different aspects of nanotechnology. One of the most ubiquitous functions of proteins is the catalysis of chemical reactions. Catalysis is a term that is used to describe what needs to happen for a reaction that is not spontaneous to proceed. Proteins are really good at taking unreactive molecules and making them do something special. They do this by binding molecules in special pockets where they can bend the molecule to put it under stress and break a bond, or they can use one of their own functionalities to cut or replace a chemical bond. And they're very clever about getting work done. Let's take, for example, the peroxidase enzymes. As the name suggests, peroxidases can break down peroxides, like hydrogen peroxide. On its own, hydrogen peroxide is pretty stable. But when it binds the peroxidase, it's cut in two, and it's made very reactive. And this is an important function that helps the immune system fight disease. In the presence of an infectious agent, for example, a bacterial cell, the enzyme converts hydrogen peroxide into a reactive molecule that will stop the bacterium in its tracks by damaging its cell wall. So this type of enzyme and the chemical catalysis that it facilitates has an important function that keeps us healthy. And there are actually a variety of diseases linked to high and low peroxidase levels. Too much peroxidase seems to be linked with coronary artery disease. The peroxidases may help to speed the formation of plaques that are very harmful. But too little peroxidase is linked with immune suppression, also problematic. A very interesting potential new role for peroxidases has been identified that is relevant to the use of nanomaterials. The enzyme peroxidase seems to be able to break down and biodegrade carbon nanotubes. And we'll talk more about carbon nanotubes when we discuss drug delivery, and you'll appreciate why this discovery was so important as we talk about how powerful carbon nanotubes can be in an in vivo system. The discovery that carbon nanotubes could be broken down with peroxidase was made by Alex Starr, a professor of chemistry at the University of Pittsburgh. Alex worked with carbon nanotubes for several years, and he first developed very powerful sensing systems using these materials. But in the course of these studies, he began to wonder what would happen to nanotubes once they were inside the human body. And he was then the first person to observe that a peroxidase called myeloperoxidase could degrade a nanotube. He first showed that this was possible in a test tube, and eventually showed that it could also occur in a living animal. And this finding really captivated the nanoscience community because it indicated that nanomaterials could be biodegradable. And again, we'll come back to this, but we'll discuss how nanotubes can be used for drug delivery, and you'll realize that if they were not biodegradable, this type of application just wouldn't be possible. It would be very problematic, because if nanotubes were left behind once a drug was dispensed, they would eventually accumulate and they could cause inflammation. And there have been studies of mice where mice that inhaled carbon nanotubes developed severe lung inflammation and went on to suffer from lung fibrosis. So this was a serious concern, and this is why finding a way for nanotubes to be biodegradable was a major breakthrough. The way that peroxidase degrades nanotubes is a really amazing process. So nanotubes sometimes exist as what we call multi-walled structures, meaning that there is a nanotube inside of a nanotube that is inside of a nanotube. This is very much like the Matryoshka dolls that maybe somebody's brought you as a present from Russia that are nested dolls. Now, when peroxidase degrades these materials, it does so one layer at a time. And Alex Starr and his team were able to observe this in real time. They were also able to observe that once the nanotubes were present as a single layer, so all of the outside layers were degraded, and then there was just a single nanotube left, the enzyme chewed from the middle of the carbon nanotube to break them into smaller pieces. And when I read about this mechanism in detail, it reminded me a lot of the way that my kids eat sandwiches that they don't like. So I have a 2-year-old, 4-year-old, both boys, and if I give them a sandwich that they don't like what's in the middle, they start to eat the sandwich from the outside, and they eat the bread. They eventually break one sandwich into 20 sandwiches, and all that's left is whatever was the filling for the sandwich with a little layer of bread. And that's exactly what happens to these carbon nanotubes, that they get chewed up from the outside, and then just the core is left. So this discovery really highlights an important discovery in nanoscience, the enzymatic degradation of carbon nanotubes, and it also gives us a good example of a chemical reaction that a protein catalyzes. Another important class of proteins are antibodies. So these proteins do all of the heavy lifting for our immune systems. Antibodies are able to specifically recognize foreign agents in our bodies and tag those foreign agents so that they're eliminated. Most antibodies have the Y shape that's shown in this figure. They have a stem and 2 recognition domains that are very specific sensors. Now, how do these proteins recognize threats to our immune system, and how do they work to eliminate them? So this seems like a hard problem to solve. Recognizing an unknown agent that your body has never seen before, how on earth do we do that? It turns out that what we do to cope with this challenge is to make large libraries or sets of antibodies. And every member of this set is slightly different. Once one of these antibodies finds something threatening, it gives us a signal that says that it should be amplified. And then many copies of it are made to ensure that an infection is cleared. The binding between an antibody and its target is very strong, and many molecular interactions are made that hold the complex together. As I'm sure you're aware, sometimes our immune systems can turn against us. For those of us with certain food allergies, it's the immune system and antibodies that mistakenly treat a certain component of a food as a threat. But we've also learned to make our immune systems work better. We know how to vaccinate against many diseases by injecting substances that mimic infectious agents. Using these types of strategies, we've been able to eradicate many terrible diseases. We also know how to produce highly specific antibodies in large quantities so that we can exploit their binding properties for medical applications. There are several antibody-based drugs out there that are able to find specific proteins on the surfaces of cancer cells, and they're able to selectively eliminate them, just as if they were a bacterial cell. And these types of drugs are very effective at controlling cancer. We'll find that antibodies are also a very powerful tool in nanomedicine. We'll look at several examples of antibody-functionalized nanomaterials, for example, metal nanoparticles or quantum dots, with an antibody tethered to the surface. And this is a way to take a nanomaterial and make it very specific for a certain type of cell or a certain type of molecular target. Let's look at one example of this type of antibody-mediated targeting that relates to the gold nanoparticles from our last lecture. It turns out that antibody gold nanoparticle conjugates are already used as indicators in many of the commercially available pregnancy tests. These are tests that can provide a result simply by introduction of urine onto a dipstick. The urine is drawn up into an analysis chamber, and it reacts with two strips of material enclosed within the dipstick. One is a control, so as long as urine was introduced onto the dipstick, a line is formed. The other indicates whether or not a hormone associated with pregnancy, HCG, is present. The outputs for this type of test are just red lines. You just read it by eye. And again, in many of these commercial tests, these red lines are formed with gold nanoparticles. The way this works is that an antibody that specifically binds the pregnancy hormone HCG is modified with a gold nanoparticle. And then there are also larger spheres in the mixture. If HCG is present, it binds the antibody and a large complex is formed. This complex then flows along the dipstick until it reaches the test site. At this site, a filter will stop the large complex from flowing through it, and it concentrates it into a line. And then close by, any leftover gold nanoparticles that are still there are trapped by a finer filter, and they're used as a control to show that the sample was actually transported through the device, so that you always have a positive signal even if the pregnancy test is negative. So here, the intense color and the nanoscale size of gold nanoparticles are being leveraged to make this type of diagnostic testing easier and more sensitive. If it was not for the nanoscale size of the particle, the filter-based approach would not work. So small molecules that we usually use as colored dyes are difficult to stop with filters. So this just wouldn't work if all we had to work with was a dye. So now you're a little bit more familiar with antibodies, and you can see how they can help a nanomaterial recognize a target molecule and sense it. And we'll look at many other examples of materials functionalized with antibodies as we go along through the next few lectures. Now, not all proteins have functions that we consider active. Others are passive, and they're present to provide a structure to a cell or a tissue. For example, collagen, which is a protein named after the Greek word for glue, is a main component of connective tissue, and it represents 30% of the total protein in our bodies. It has a very interesting structure, so rather than being variable, it's quite constant, and it always exists as a triple helical structure about 1.5 nanometers wide and 300 nanometers long. These triple helices of collagen are wound into tough bundles. They can be hundreds of nanometers thick, and they provide a guide that can tell cells where to grow. And there are a number of diseases related to problems with collagen. So I'm sure that you've heard of scurvy and know that this results from a deficiency of vitamin C. Scurvy was at one time a serious issue for those on long military expeditions at sea, but it is fairly rare now that its origin is well recognized. It turns out that the vitamin C deficiency that causes scurvy produces defective collagen, and the connective tissues are affected. The gums in our mouth are particularly susceptible to this, and tooth loss was a major ramification of scurvy. As well, healing of wounds is a process that's expedited by collagen, and slow healing was a major symptom of scurvy that made people affected with this disorder susceptible to infection. Collagen is an intensely studied protein also because of its role in our physical appearance. So the changes that appear in our skin as we age, those come about because of collagen breakdown. So as we age, the collagen in our face and other places kind of breaks down and we lose the structure of our skin. So many cosmetic procedures have been developed that attempt to reintroduce collagen to repair the effects of aging. Collagen is also an important component in types of artificial skin that are under development and an important ingredient in treatments given to burn victims. The relationship between collagen and nanoscience is that this protein has been used as a model for the development of nanomaterials that can support cell growth. As we just discussed, collagen is oriented in fibers, and its directionality and the spacing of the fibers tell cells how to position themselves. And this natural phenomenon has been mimicked with nanomaterials. Researchers at the Massachusetts Institute of Technology have produced nanoscale gratings that mimic the structure of collagen. And they found that these gratings are effective at orienting cells. So, for example, here you can see a cell that is growing along a nanograting. Without the collagen mimic, the cell simply spreads. It doesn't have any directionality at all. And it turns out that it's very important for cells to have directionality in order to be able to build up tissues and organs in an organized manner. And so these types of materials have tremendous promise for artificial tissue engineering. We'll return to this topic in a few lectures. For now, we're talking about the different functions of proteins at the nanoscale, and we've talked about 3 different functions so far. We've talked about proteins that can catalyze chemical reactions, proteins that are good at recognizing other molecules, and proteins that provide structural support to tissues and cells. So now let's look at one other important function of proteins that relates to a role that they can play in nanoscience, and the role that they play as cellular gatekeepers. Now, cells are very picky about what they take in. They do this to protect themselves. And it's rigid requirement that very delicate balances of different molecules be maintained in cells. And there are proteins that sit on the surfaces of cells, and their function is to decide what should be allowed into the cell or what should come out of the cell. These proteins, and we call them receptors, are inactive if they don't have anything bound. They just kind of sit there and wait for something to happen. But once they do bind something, they trigger an amazing chain of events that causes the cell to make pockets in its outer membrane. And these pockets, if you imagine the cell membrane and it's nice and smooth, and then this protein binds something that it wants to take in, and then the membrane will just loop in, it'll make a pocket, and the edges will reclose, and then whatever the receptor wanted to drag into the cell comes in. This type of process plays an important role in how cholesterol gets processed. One type of cholesterol is packaged in a nanoscale particle formed by a protein called low-density lipoprotein, or LDL. These particles can bump into a receptor on the surface of the cell, and the cell takes the LDL in. Now, LDL is what we think of as bad cholesterol, right? That's the one that you don't want to see showing up when you go to your physician for a checkup. But there's a reason for this. So if we don't get that LDL into cells, it's running around in our blood, and that's when it can do bad things. If this process goes smoothly and the cholesterol is taken into the cell, then it can be metabolized and processed very efficiently. So individuals that have a defect in one of the proteins that sense the cholesterol particles, the LDL, if you have a defect in that, you will be very susceptible to having high serum cholesterol concentrations, and you'll be more susceptible to the coronary and vascular issues that come along with that. Now, LDL is roughly 15 to 25 nanometers in size. And so the cell is used to seeing nanoparticles of this size. And interestingly enough, it turns out that gold nanoparticles can use the same pathway to enter cells. They bind receptor proteins on the outsides of cells, and then they're taken in through these pockets that can be formed in the membrane that I just described. And how efficient the uptake is depends quite a bit on the structure and the size of a particle. A colleague of mine at the University of Toronto, Warren Chan, studies this issue in detail. Warren actually started his career using nanomaterials as medical imaging agents, and we'll actually talk about some of his work in the next couple lectures. Recently, he's turned away from using nanomaterials as imaging agents and turned over to looking at the details of how nanomaterials interact with cells. His lab has incredible capabilities when it comes to studying very specific and detailed cellular phenomena. but he also has significant experience in the development of interesting new nanomaterials. Recently, Warren's group did a study where they looked at the uptake of gold nanoparticles and nanorods into cells. What they found was very interesting. While the spherical particles that they studied, and these were particles with many different sizes, they all entered the cells. But when they changed out the particles for rods, they did not see any uptake into cells. So it seems as though a cell can be fooled into thinking that a gold nanoparticle is a particle of something else, like cholesterol. But it knows that the aspect ratio on a rod is not right and that that material does not belong in a cell. The Chan group also observed that the closer the size of a nanoparticle was to something biologically relevant, like LDL, the more efficient the uptake was. Now, how did they do this study? How did they tell whether or not the gold particles were getting into the cells? Well, the Chan group uses quite a bit of a technique that I think you've already heard about called scanning electron microscopy. So they incubate the material with the cells that they want to study, usually human cells that you can culture in the lab. And then they take them over to the electron microscope, and then they actually image the cell. They look at different pockets of the cell. They look for the gold nanoparticles stuck in the membrane, or maybe in the cytoplasm of the cell. That's just kind of the stuff in between the organelles, or maybe even inside the nucleus or other organelles like mitochondria. And they count up how many particles are there and look at the localization profile to draw some conclusions about whether or not the particles could get into the cell. And so it's a very precise measurement that they use to study this phenomenon, but as you can see, it gives us a tremendous amount of information. This type of study is quite helpful as new nanomaterials are being designed for applications that take place both on the inside and the outside of the cell. We can use this information. We now know that if we want a nanomaterial to enter a cell, let's say perhaps to deliver a drug, then it should be spherical and as close to the size of a natural nanoparticle as possible. On the other hand, if we want a nanomaterial to stay on the outside of a cell, perhaps to tag the cell as a cancer cell for detection, it should be rod-shaped. We will return to how receptors can be used with nanomaterials in the next few lectures. In this lecture, we've reviewed the main functions of proteins, and we've linked each of these functions to the world of nanoscience. Now that we have all of this information about proteins, and all the information that we went through in the last lecture on DNA, we're ready to talk more about specific applications in nanomedicine. In the next lecture, we're going to discuss a cutting-edge approach to detecting cancerous tumors in the human body. And we'll talk about how sensors made of quantum dots can serve as beacons that will illuminate sites of cancer. See you then.